And welcome back, everybody. Happy to announce our next presenter, Enzolytics. It trades on the OTC Pink sheets under the symbol ENZC and is a drug development company committed to the commercialization of its proprietary proteins for the treatment of debilitating infectious diseases. Joining us is the CEO of Enzolytics. We have Charles Cotropia. Welcome, Charles.
Yes, thank you, Anna. Glad to be here. Thank you for having us.
All right, we look forward to your presentation. You may begin whenever you're ready.
Okay, thank you, Anna. I appreciate it. And thanks to Charles and Ryan for getting us ready and making this possible. I hope everyone can hear me and see me sufficiently. Got a lot that I'd like to convey and share with the audience today. Got a number of slides. I'm going to go through them fairly quickly, because I have presented quite a few here. We have a lot of information to cover. And so let me get started. Enzolytics is a drug development company, and our focus is on producing therapeutics to treat HIV and other infectious diseases. And we have a very strong, this says executive team. It's really our science team. Harry Zubilov, who is our chief science officer, a renowned researcher in the biotech field, having worked with his father, Dr. Harry Zubilov Sr., for many years. My brother, Joseph Cotropia, who has been in research for all of his adult life. Gaurav Chandra, a physician who has been with us for many, many years. Very seasoned in the development of therapeutics. And our advisory board, Dr. Moss, Dr. Bryson, and Ellen Batetta. Let me move more quickly through these so that we can cover exactly what I would like to convey to the audience. We do our development certainly with our in-house scientists and individuals, but we also work with outside entities and research institutions of all sorts. This gives you kind of an idea companies that we work with. Everyone from GenScript, University of Strasbourg in France, STC Biologics, and all the rest. So we're not alone in this. We do have partners that we work with. I want to cover three areas that we focus on, and these three areas are shown in this slide. I'm going to look to the second one first, because it's the one that is leading the way to therapeutics for the market. And that's the therapeutic of a peptide called an inactivated pepsin fragment that has been tested against HIV in clinical trials, which I'll cover, and has shown very promising, effective results. Complementary to that, and the reason the two companies, Biokinetics and Enzolytics, have together, the technology that Biokinetics brings to the partnership, and that is the ability to prepare fully human monoclonal antibodies. Now, a third component is the use of artificial intelligence. And we have on our team Denver Scientific. And the reason for that component is the necessity to identify the correct site on viruses that will permit the neutralization of the virus. And I'll get into that in a little bit more detail. So what is the problem? I'm only going to take a minute or two, but it is a problem. I was interviewed on CEO, CFO magazine a couple of weeks ago, and it's on our website, that interview. And the first question was, isn't it the case that HIV is no longer an issue? And I was a little bit taken aback, but not really, because that is the misconception. There are 38 million people living with HIV, over 1 million in the US. Only 54% have access to antiretroviral treatment. That leaves 18 million without any treatment. 1.7 million become newly infected each year, and then you can go through the rest of the very serious statistics about HIV. When you look at the treatment, I hear on the television every day the new advertisement for Biktarvy from Gilead. $42,000 per year for this therapeutic. Dovato, I hear the ad every day on the news, $27,000 per year. The total life cost of treating HIV is from $250,000 to $400,000. And the drug market is $30 billion, going up to $34 billion in 3 years. When you look up Biktarvy, here are the side effects. I don't expect anybody to be able to read these. There's so many. Worsening of hepatitis B, change in the immune system, kidney problems, kidney failure, et cetera, et cetera. Same with Dovato. The list just is enormous. And we all know from the lifelong use of these therapeutics, which are chemotherapy, not immunotherapy, they don't cure. They're very costly, as I pointed out. They cause damage to the heart, the kidney, the liver, decreased bone density. So that's the problem. Now we have the coronavirus, COVID-19, and we see around the world the damage that is being caused by this virus. Here are the statistics. World cases, 135 million. I read on the news about 30 minutes ago that they estimate that the number of cases in India are 300 million because their count is behind what is actually happening. So this number of world cases of 135 million is not accurate. The US cases, US deaths. Now we all believe that we're reaching herd immunity. We can go out, not worry about the virus. The experts tell us something different. Dr. Offit, director of the Vaccine Education Center at the Children's Hospital in Philadelphia, was on the news just a week ago. And he said, no, COVID-19, the coronavirus, is going to be with us year to year. So here's the therapeutics that we propose and that we're developing. The first is ITV1, this peptide that has been tested against HIV. And then there are monoclonal antibodies that are being produced targeting HIV. Get into the specifics. Well, the first is covered by 2 patents. The inventor is our CSO, Harry Zobolov, and we take patenting very serious, being the size company that we are. These patents are owned by the company. The therapy is one that has been clinically tested in HIV patients under the European Union standard. These were test 16. And unfortunately, because of cost problems and other things that interfered with the preparation of the therapeutic, they were not able to be completed. But now, well, let me get into what the results were. Extremely beneficial results. 80% drop in viral load, 68% increase in CD4 count, and you just go through the parameters that it is aware of. Very effective therapeutic shown in those clinical trials. We are now preparing to conduct renewed clinical trials, but not under the Bulgarian Drug Administration because it has been supplanted by the European Medicine Agency. So we have a new agency that we're going to be testing the product under. How will we achieve that? We're entering into a partnership with European partners where we will be the 50-50 owner of the partnership. We will provide the therapeutic for the initial validation, and then other European partners will actually produce and actually pay for the clinical trials. A very significant finding for the company to be able to find partners who will actually be funding the clinical trials. The therapeutic would be distributed in Europe and Asia. And because the EMA and the FDA are now under a EU recognition treaty between the two, approval in the EMA may lead to FDA. So that's the first therapy. The second therapy, monoclonal antibodies. What are they? We all really, and maybe we don't know exactly what they are, but our body produces millions of these proteins to fight any virus and any bacteria that the body confronts. The flu, a bacteria, even HIV. Unfortunately, HIV is stronger than the body's antibodies that are produced. 1% of those that get HIV are long-term non-progressors and do produce an antibody that can control the effect of the virus. But for the others, the body doesn't control the virus. We're also aware of monoclonal antibodies because we know and have heard that President Trump was administered 2 produced by Regeneron against the coronavirus. Eli Lilly has produced 1. Experts agree that you need a combination, and you also need antibodies that target a site that does not mutate. The NIH experienced this mutation problem when it produced anti-HIV monoclonal antibodies called VRC01 and VRC02, and after some years of testing, reported that it was not effective. And the reason it was not effective was what the Institute called viral escape. That means the virus mutated around the monoclonal antibody. So what do we have? This is just a slide that says convalescent plasma is also attempted to be used, and it's a use of antibodies made in a person that has overcome the virus. But convalescent plasma doesn't give you exactly what you need. Our technology is to produce targeted monoclonal antibodies that address the specific Achilles heel of a virus. Now, let me take you through this. This is the heart of what we do. First, to identify an immutable, conserved site on the virus. Otherwise, you're producing something just like the NIH produced with VRC01 and VRC02 that will target an area that mutates. Then you have to produce, we do, a human-based monoclonal antibody working from peripheral blood from a person who has had the virus and has prevailed over it. You produce the virus, third component, and then you improve it by ways that are capable of improving monoclonal antibodies. Let me get into this a little bit more specifically. This is the HIV virus. It's part of it. It's GP41. Total virus is twice this many amino acids. The objective and the necessity is to identify the immutable site on the virus that is always there. And how do you do that? If you have the capability of doing computer analysis, which Denver Scientific has and is our partner. You can survey or curate the 87,000 difference of the HIV virus. In other words, this thing has changed slightly 87,000 times. If you do the computer analysis, you can identify those sites where the virus has not mutated. In other words, this sequence 1, 99, almost 100% conserved over all 87,000 viral clades. Then you produce a monoclonal antibody to target that site. Now, how do you do that? That's the key technology. It's not easy to do, I can tell you. And my brother has the job of doing it, but he has done it with Clone 3, an anti-HIV monoclonal that has been tested in each of these 5 labs around the world. And what was the result of the test of that antibody? It neutralized 95% of all the various isolates against which it was tested. And those isolates are those located in regions around the world. Now, how do we know that was effective? As we did the computer analysis, it turned up in one of these that was in the 98% range of conservativeness for the geometry of that virus, of that monoclonal antibody. And that's the same thing you want to do with regard to each of these sites on the virus. Quite a task, but it can be done, and that's what we're doing. So knowing that you can do it for HIV, it can also be done for the coronavirus and many other viruses. This is just the steps that we had to go through for Clone 3, the one antibody that I just gave you. All the steps to get you to the point of having the monoclonal for testing. Now we're at the test where it's being finally tested at the University of Strasbourg in France. Animal trials will be at the California Primate Research Center and then clinical trials. But here's a significant point about our technology. Once you've done it for HIV, and we know that there are correlative structures, HIV and the coronavirus, that means there's similarities. You can do the same thing for the coronavirus. How do you do that? This is the coronavirus. It has amino acid sequences similar to the HIV virus. You have to analyze, and we have the 50,000 variations of this composition of amino acids, and you identify those sites that are always there in each of the 50,000 variations. And why do you do that? Because when you prepare a monoclonal antibody that targets this site, the first one, sequence 1, you want to make sure that that site is going to be there in the next variant. Because if it's not, your monoclonal antibody will not be effective. So you produce monoclonal antibodies against each of these. And you have a cocktail. As President Trump was given a cocktail of 2, you might have a cocktail of 3 or 4. So the focus is to prepare monoclonal antibodies against sites that are always present as the virus mutates. The sites we have identified are in existence in the variants that are now plaguing not only the United States, but the world. The UK variant, the Brazilian variant, the South American variant. They're included in these sequences that we have identified. And we have filed patent applications on the sites that I've just identified. And why do you do that? Because a patent owner has the exclusive right to make, use, sell, import the technology patented. And we think that's very significant, not only the epitopes for the site, but the monoclonal antibodies that are being produced targeting those sites. We believe that's very significant, certainly for a company that is not as large as the big pharma. So this technology not only can be used for HIV or the coronavirus, and we have used it for these over at the left, rabies, influenza, influenza B, tetanus, and diphtheria. They have not been commercialized, but they've been produced. You can also produce them for all the ones listed on the right. The list goes on from HIV-2 all the way down to the bottom, including viruses that affect the animal kingdom. For example, all of these, including the elephant endotheliotropic virus that is plaguing the elephant species. So once you've done all that, then the objective is treatment, and that's our focus. There's also a monetary advantage. We had a consulting company analyze from a financial standpoint what is the likely earnings and over a 10-year period using North America, Europe, Asia, and Asia as your market, and assuming a penetration from 2% up to a maximum of 20%, the expected earnings would be $100 billion in the first 11 years. So I've taken a lot of time. That concludes my presentation. I'd be glad to take some questions from our viewers.
Great presentation. We have so many questions that we will absolutely send to you, Charles. So many questions, but we only have about 5 minutes left. So let's start here. When do you expect testing in the U.S.?
Well, we're going to do testing first in the European Union, and that testing will be occurring. The production will be in the next few months. Testing will begin immediately after that. So clinical trials will begin and will be completed, hopefully, within a year to a year and a half, and then we will be ready for market. So that's the timing for trials in Europe.
And what is the timeline
Europe
for the completion? What about the completion of the clinical trials? When are we expected to start those trials and complete them?
Clinical trials will begin probably in about 4 or 5 months, depending on what happens is we prepare a validation order because we have the therapeutic ready to go. And then once the validation order is completed, then clinical trials would be commenced. And we have a CRO preparing the protocol for clinical trials as we speak.
Who are the European business partners that you're working with?
They're European principals that we've known and that Mr. Zobolov have known for years. They're people who were involved in the earlier development. So they're people that know the therapeutic, they know the capability of it, they know the results of the earlier trials, and they're principals that are just known to the company and believe in it and are willing to, and I might say that those principals will be financing the clinical trials once we produce the therapeutic that will be used. That's a tremendous financial benefit for the company. I mean, tremendous benefit. They believe in the technology and they would benefit from having the technology make it to market.
Do you see any pharmaceutical companies that will license your patents in the near future?
We hope. We have, over the years, and even as we speak, we have always been open to Big Pharma. They've always encouraged us. They've always said, as soon as you get to a certain level that we agree with what you're doing, we will be interested. So yes, we have to prove it. And that's what we're doing. It will be proven. And given the magnitude of the capability, and you notice the number of viruses that could be addressed, yes, a large company for implementing this would be optimum. And that certainly is in the cards, and we would welcome that.
Will these partnerships affect stock price?
Well, no, if we have a partner, it will affect stock price, and I think it will be favorable. I can't see it being unfavorable, because you have to look at the market that is out there. $100 billion, Gilead and Merck's net profit over the last 10 years was less than $100 billion. So it's a tremendous market. And so if it affects stock price, it's got to be positive. And I don't want to project what it might be, but it's if you have a successful therapeutic for this market, it's going to be positive for everybody involved.
Are you trying to get the company to get emergency usage or FDA approval? Is that on the horizon?
We haven't taken that road yet because it is a very quick road under the EMA. And that is, in the next couple of months, we will have a validation order for what has been previously tested. And then human trials, clinical trials, will begin thereafter. So that has to be done. And we haven't really approached it from the FDA standpoint to date. Certainly, there is a combination, a recognition agreement between the FDA and the European Medical Agency, which might allow, once it's approved under the EMA, to be approved under the FDA more quickly. But we're focusing first at the European side because it's more quickly, it can more quickly be accomplished.
Well, Charles, you've done a great job. There's so many questions here. I wish I could read all the answers that you're going to send to the viewers, but I do want to give you a little feedback. A viewer has said, thank you so much for everything you and your team are doing. My question is, do you and your company know how much your investors love and support all your hard work? From the bottom of our hearts, we do. So there you go.
That means so much. We appreciate that. And we hope to be successful for them and for the health of the world.
Absolutely. Thank you, Charles. Really looking forward to all of your updates. Thanks for joining us.
Okay, we'll give them. We will be giving them. Thank you so much. Thank you, Anna.
Everyone. Yes. What a great day. We have one more presenter coming up. So as you know, you're going to see a black screen for a brief moment. Stay with us as we bring on our last live presentation. We'll be right back.